Next up, I am pleased to present GenSight and President and CEO Bernard Gilly, who's going to give us a few minutes of prepared remarks.
Thank you, Bernard. Thank you. Well, thank you very much for inviting me. I'll just spend the next few minutes to introduce you as GenSight. So this is this thing. So we're a phase 3 stage gene therapy company. We have essentially two programs that are dealing with retinal disorders. The first one is based on a technology platform which enables us to deliver missing protein in the mitochondria, which is really a proprietary system that has no equivalent. And the second technology is Optogenetics, which is better known, and we are probably amongst the first to apply this for therapy, and we are about to start the phase 1 in retinitis pigmentosa patients. So our most advanced program is GS10 and is targeting Leber hereditary optic neuropathy, which is a clearly devastating disorder. In terms of the next milestone, so as you see, GS10 targeting LHON is now well into phase 3. We are completing the recruitment of the second trial, and we are going to announce the results by Q1 of next year, meaning that we'll be probably less than 2 years from the product approval, and we are starting the launch readiness activities. Whereas the second most advanced program, GS30, is now completing the GLP tox model in tox trial in monkeys, and will move before the end of the year in RP patients, and we're expecting quick results out of this. The technology around the mitochondria is depicted here. It's a unique technology that allows to put the missing mitochondrial gene into the nucleus and allow the messenger RNA to 100% get to bind to the ribosomal complex on the surface of the mitochondria and then to the protein to be internalized. And we're treating LHON. You probably have heard about LHON. It's a very sudden, brutal disease that happens in young adults or late teens, 15 to 35. And those patients are losing sight in the two eye in a matter of weeks to months, and 97% of them are blind by the end of the first year of their disease. And these are the results for a phase 1/2 trial, and these are the patients that are less than 2 years that have been treated in one eye, which, by the way, was the worst eye. And as you can see over time, and we have now 78-week data over time, the improvement of visual acuity is more than 4 lines ETDRS letters equivalent in the treated eye as opposed to the non-treated eye. The optogenetic technology is quite straightforward. We're using a bioengineered AAV2 that we have been licensed from Adverum, and we're using Crimson R, which is a red-shifted channel rhodopsin, which has the kinetics that suit well with the vision. And we have developed this Optronics device, which are the form of goggles, that in fact transform the visual information into red-lighted visual information to be sent to the transduced cell in the retina. And this is the result, the efficacy result. This is the monkeys. And as you can see, we have a nice light intensity dose response curve in these monkeys, which is maintained over time, which is what we were expected, and a nice transfection rate through intravitreal administration with the AAV278. Thank you.
Thanks, Bernard. Maybe going back to GS10 for loans. It looks like there's a little bit of a placebo response. Maybe just comment on what's going on there.
No, I don't think we'll call this a placebo response. I think we figure that in low vision patient, the variability of visual acuity measurement is quite high. In normal individual, it's between 4 to 6 letters. In low vision individual, in low vision patient could get to 13, 14 letters. So I would rather consider this as part of the variability in the non-treated eye.
And it looks like, from looking at the graph, that the data gets better over time. Are you guys going to be updating the data set later this year, or what's the plan for this data set?
Yes, we'll release the 96 weeks, the 2 years data in June next month. And we expect that the difference is going to be maintained over time. I think it takes time to probably restart the mitochondrial machinery in those patients, But once it kicks in at 24 weeks, then it's deemed to be steady state, and then we're probably getting to a plateau in between 48 to 78 weeks. I believe that 96 weeks data will just confirm that we're getting to the plateau of response. Again, this is a phase 1/2 trial, so these are very heterogeneous patients and patients that are, so I think we have only one or two patients that are less than 12 months in their disease. So they are very severe patients here.
Got it. And maybe just walk us through some of the differences in the phase 3 studies versus the phase 1/2.
Sure. So based on the data from the phase 1/2, we decided to conduct 2 parallel phase 3 studies. The first one is RESCUE and is recruiting patients that are 6 months into their disease, so less than 6 months since the onset of vision loss. And the second trial, called REVERSE, is addressing patients that are between 6 and 12 months. The idea is really to treat as early as possible and maximize the chance of success to either maintain vision or eventually restore part of the vision to these patients. We have completed REVERSE recruitment already by the end of February. We are about to complete RESCUE recruitment. And then with the agency, we've agreed on the 48 weeks follow-up on these patients, which will be Q1, end of Q1 18.
And what are the studies powered to show, and what are the main endpoints you're going to be evaluating?
So the primary outcome, as discussed with the agency and agreed with the agency, is a difference in visual acuity as compared to baseline between the treated eye and the untreated eye. FDA requires at least 15 letters different, and we have powered the study at 85% to make sure that we match this. We are also following several secondary outcomes on these studies. We are following the number of the percentage of responders. We are also measuring the quality of sight and the quality of life of these patients in order to prepare for the discussion with the payers.
And you mentioned you're treating just one eye of the patient. Maybe walk us through the rationale here for this approach.
So that goes back to a number of discussions with the agencies, both in Europe and in the US. Obviously, the preferred route for the agency would be to randomize patients. The issue with this is that we and the KOL with whom we are working and the principal investigators consider that indeed the sooner we treat after the diagnostic, the better we'll be. And we may also see patients losing their chance of restoring vision if we wait too long. So randomizing patients will mean that we'll inject bilaterally in one patient, and the next one will have only sham or placebo. And it's only 1 year after that we may eventually do a crossover. So that for us and for the API was not acceptable. So we went back and forth with the agencies. They finally accepted that we randomize the eyes instead of randomizing the patients, so that at least all patients get a chance to be treated. And we also have committed to a bilateral study that is going to start in September, which will be used as a supportive study for the approval.
Just given that you're capturing patients early who have just have disease onset recently, should we think about this more as an incident market opportunity, or is there some prevalent aspect we should think about as well?
For us, it's an incidence model. There's no question about this. We will know from the 2 phase 3 whether we can go slightly more into a prevalence model. But I really strong doubt that after 18 months of the disease, we can really restore the mitochondrial activity.
I think you touched on this in the presentation. Maybe taking a step back, the ability to target mitochondrial DNA is pretty unique, and maybe just give us a little bit more on the technology that allows you to really address this versus your more traditional nuclear DNA.
So that goes back to a lot of science. I'll try to do this simply. Mitochondria are, in fact, the powerhouse of the cells, and they resulted from an ancestral infection of a bacteria into a cell. And over the evolution, this bacteria has transferred most of its genome into the nucleus of the cells, but there's still a number of genes that remain in mitochondria, and particularly those who are important for the respiratory chain complex. So the idea that was developed by our scientific founders was to use the mechanism by which the mitochondria are getting their messenger RNA from the nucleus, and use this technology to allow for providing the missing gene, the missing protein into the mitochondria. And once we're able to do this, it's really a virtuous circle for the mitochondrion, because you know that we all are heteroplastic for mitochondria. But now, even if we have a population of so-called bad mitochondria that continues to proliferate in our cells, we don't really care, because they will find the missing messenger RNA in the nuclear periphery, and they will be able to get the protein.
Maybe switching gears to GS30, the Optogenetics program. Maybe talk about how gene therapy differs from some of the other optogenetic protein approaches, and your selection of the light sensing protein.
So we really paid a lot of attention to this. The most simple way to, and that was the work done by Hans Bamberg in the early 2000s, was to use a channel rhodopsin, a blue lighted channel rhodopsin. The problem with this is that blue light is quite toxic for the retina. It's been described already and published, and also it's very difficult to play with the intensity of blue light, because as you need to increase intensity, then you'll be eventually doing more harm to the retina. So we were trying to find a channel rhodopsin that would be shifted to a larger wavelengths, and when we finally found this into Ed Boyden's lab on the other side of the river at the MIT, and that's really great, because this crimson R is not only redshifted, but it has a kinetics that is consistent with vision. We need this protein to react very quickly and to shut up very quickly, because then the next vision event can happen in the same cell. So we need to have a kinetics that will be below 15 milliseconds, eventually as close as possible to 10 milliseconds, which is the case of this crimson R. So with this, we have the opportunity to develop an optogenetic approach that is indeed working exactly like the rhodopsin, same time, and the whole game was to transfer this into a cell that is still functional, and we choose the ganglion cell, because ganglion cells are there, and in RP patients, as well, by the way, as in later GA patients, the ganglion cells are still alive and functional, and they're used to send the message to the brain. So the idea is to give them the ability to create themselves the electric signal and then send the information to the brain, which is what you've seen in the monkeys.
It's pretty remarkable that you can essentially turn a retinal ganglion cell into a photoreceptor and have the brain interpret that and see again.
Sure, and the pioneering work of this were done by those guys who have developed artificial retina, because they are delivering electricity, but in fact, once the electric signal is sent by this electrode array, whatever they are, then that triggers the ganglion cell to send a further signal to the brain. So we're really piggybacking on this. They are using a tissue stimulation, because electrodes are really just delivering a huge amount of energy. We are more at the cellular level.
Any questions from the audience? And when do you expect this approach to enter the clinic?
So we will file the MHRA in UK and the French agency in September. We expect to be first in man by December of this year, and we are going to use to recruit RP patients, which means that soon after, probably no later than mid-18, we'll be able to report first efficacy proof of concept in these patients.
Great, and I think that's all we have time for. Thank you, Bernard.
Thank you very much, Steve.